This is part 6 video. So as I have told you all that you maintain a practice copy. So I myself maintain a practice copy for me, and also I have a notes copy. So if you all message in my given number in the description of my part 1 video, so you can just go there and message me. I can send the PDF of my notes copy, but in this YouTube I upload the videos of my practice copy, how I am doing it, and explanations. Also I give some screenshots of my note copy, but if you want the PDF of my note copy, then you can message me. So this is my aldose family tree, and I have just drawn it for practicing some questions of diastereomer, epimer, anomer question. Anomer I have not started, anomer I will start today. Epimer questions I will practice with you. So over here the epimer question I have written glucose versus mannose. So what you need to do, you need to remember the structure of glucose, you need to remember the structure of mannose, and as you remember the structure of glucose and mannose, you can say what epimer it is. So glucose is this, mannose is this. So you can clearly see that there is a change in the C2, carbon 2. So they are C2 epimer. So they are C2 epimer. Okay. Okay, next question is allose versus glucose. Allose versus glucose. So this is my glucose, this is allose. So you can see that there is a change in the carbon 3. So they are C3 epimers. So this way you need to do the questions. Sorry, glucose versus galactose. So you can see glucose versus galactose over here. So they are, you can see a change in the carbon 4. So they are C4 epimer. D-glucose and D-mannose, again the first question only I have repeated. So they are C2 epimer. So you can see it. So this way you need to do the epimer question. You need to practice the structure, remember the structures. If you can't remember the structure, remember the table. As you go in the exam hall, as soon as possible try to draw the ketose family tree and aldose family tree. All the questions will get easy from that. Okay. Now we are going to start anomers. So let's check what are anomers. So we have understood that epimers are C2, C3, C4, but diastereomer also have another type, that is anomer. Monosaccharide structure convert from linear structure to cyclic form. So this monosaccharide structure, you are converting them from linear structure to cyclic form. This is called anomer. Okay. Monosaccharide structure, that is Fischer projection, convert from linear structure to cyclic form. So they are called anomers. In aqueous solution, sugars tend to form cyclic structure rather open form. So in aqueous solution, these sugars tend to form cyclic structure rather than this form. Ring form and energetically more stable than open form. Ring form are energetically more stable. So these ring forms, anomers, are more stable than this linear form. How linear form? Cyclic form in aqueous solution. Linear form, how this are done in ring form. Cyclic form in aqueous solution, ring formation occur by reaction. Aldehyde or ketone react with an alcohol, form hemiacetal or hemiketal. So aldehyde or ketone react with an alcohol, form hemiacetal or hemiketal. You can see. So this is how anomers are formed. Now just I'm explaining you, then I will draw in hand and show you. Okay. So these two are the reactions how anomers are formed. So what we understood from this, that monosaccharide structure converted from linear form to cyclic form is called anomers. In aqueous solution, sugars tend to form cyclic structure. Ring form are more stable than open form, energetically more stable. Ring formation occur when aldehyde and ketone react with alcohol to form hemiacetal or hemiketal. So reactions you need to understand how it is happening. So let's see. So basically, aldehyde or ketone react with alcohol. Can you see? Yes. To form hemiacetal or hemiketal. Let's see the reaction now. First you take an aldehyde. It's like this. It reacts with OH. What it is form, it is saying, just understand and do it. R. So C, OH it will happen, and another H, draw it here. So what will get attached? OR dash. Isn't it? This OR dash will be remaining. This H will go with OH. It will react and another H will come this side. So OR dash will be left. When I talk about hemiketal, this is hemiacetal. Sorry, hemiacetal. If I do the hemiketal reaction, then it is okay. So this is also like this. OH, OR dash, isn't it? This is hemiketal because this will take a double bond here. So this is hemiketal. Now hemiacetal and hemiketal in sugar. Okay. This hemiacetal draw the structure of glucose. Then I will explain it to you. CH, HOH, OHH, HOH, HOH. This is CH2OH, isn't it? Okay, this is your glucose structure. Now, if I convert it into hemiacetal, try to understand. If I convert it into hemiacetal, there are two possible structures. One is pyranose, another is furanose. Pyranose is more dominant in glucose and this is less dominant, but if it is pyranose structure, then the C1 and C5 OH, this OH are affected, and if it is furanose structure, then C1 and C4 OH are affected. Okay, so these two, anyway, pyranose structure, just C1 how this is done. Try to understand. Okay, so this is your pyranose structure and furanose structure is like this. Okay, so these two, just you have gotten basic idea. From hemiacetal, two kind of rings are possible. One is pyranose and another is furanose. Pyranose is more dominant in structures like this glucose and furanose is less. When pyranose, then C1 and C5 alcohol group is affected. When it is furanose, then C1 and C4 OH group is affected. Till here it is crystal clear. Okay, so now let's try to learn how you can convert a Fisher projection to Howard projection. Focus over here. I will teach you everything step by step. First, draw the structure of glucose in Fisher. Fatafat. Take a pen and pencil and just do with me. Okay. So, as I told you, in pyranose structure, we will do pyranose. In pyranose structure, C5 and C1 is affected. So, what will happen over here? This will be affected. So, one H will go here and this O will come here. The structure will be like this. CHOH. Just take it down. HOH, OHH, HOH, H, like this. So C, just more nicely you can write it as and write the H this side, OH this side. OHH, HOH. So this is your pyranose structure if I write it nicely, okay. So this way you can convert into pyranose structure. Now if this is on the right side OH, then it is alpha D glucopyranose. Understood? If this OH is on this side, if you write, then it is beta D. Understood? Now you can draw. Now as I move on, okay, you will have to now convert it into this ring structure. This much you have understood clearly, okay. Now let's draw the ring and try to place this. If I take the ring like this, draw the bonds. Now how you will place this? If I give you a simple technique, that left side group will go in top position, right? Left side, this left side group will go in top position. Similarly, right side group will grow in the bottom position. Okay. Now very easy. You start marking carbon 1, 2, 3 first. 1, 2, 3, 4, 5. Over here the 6 will come in any one. So 1 is this. Left side is H, right side is OH. H, OH. Second is this. Left side is H, right side is OH. H, OH. OH, H. OH, H. H, OH. Now your question comes to you, this and this, between this and this, put the bulky group in the top, so CH2OH, and the light in the bottom, okay. So this way you have completed this structure. Understood? OH, H, OH, OH, H, HOH, CH2OH. Now as I told you that if OH is in the bottom, then it is alpha, means first OH is in the left side in right side in Fisher, then it is alpha. So if OH is in the bottom in the pyranose structure, then it is alpha D glucopyranose. And if OH is in the left side over here, okay, left side of this, means OH in the top, then it is beta. So just take your time and draw beta D glucopyranose for me. Okay, you can do it. Draw this one also, then draw this one. Okay, entire process, because first as you practice the entire process, then only you will be able to do. Okay. So I hope you all have taken your time, drawn it. So let me now show you those who have not done it or unable to do it. Okay. Draw the structure of glucose first. See, chalo, do with me, tell and do with me. Okay. CHO, HOH, OHH, HOH, HOH, CH2OH. For more problem, if you are unable to understand, carbon 1, 2, 3, 4, 5, 6. Okay, okay. Till here understood? Okay. Now we are making pyranose structure. So C1 and C5 will be affected. Okay. This H will go here and O will be coming here. So just do it. CH, O, H. You can see. Okay, okay, okay. H, OH, OH, H, H, OH, H, and this O is going here. CH2OH. Just draw this nicely. Beta means if OH is on the right side, okay, then it is alpha. If OH is in the left side, then it is beta. So we'll do this OH in the left side. Chalo. If we are doing beta, okay. So let's draw the structure then. OH in the left side means beta. C, OH, H, H, OH, OH, H, H, OH, H, okay, CH2OH. We have done. Half work is done here. Understood? Now we convert it into pyranose structure. So like this, pyranose. I crooked the lines more nicely. Let me draw. Okay. Now draw the bonds. This is carbon 1, this is 2, this is 3, this is 4, this is 5, this is 6. This is 1, this is 2, this is 3, this is 4, this is 5. Okay. Beta. Left, all the left on the top. So OH, H, H, OH, OH, H, H, OH, bulky on the top, CH2OH, and light on the bottom. So this is your beta D glucopyranose. Just if you are forgetting, then remember one thing. OH in left, mark this, OH in left is beta, that is OH in top in pyranose structure, and OH in right is alpha, that is right in the bottom, isn't it? Right in the bottom. So that is OH in bottom. This is your conclusion. Understood? So this way you can just go on beta and alpha structures, pyranose. I hope you have understood how you are doing. Next we'll practice one more structure that we'll do in the next class. Thank you.